All right. Welcome back, everybody. Sorry for the technical challenges. My name is Matt. Today we'll be going over our May Tidy Biology virtual meetup. Today we'll be doing some pathway analysis, and Akshay will be joining us in a little bit to do a live code along, taking an RNAseq data set all the way through to pathway analysis and some data visualization. But before we get started, I have a special guest with me today. So Avi Maayan from Mount Sinai Bioinformatics Center is going to be telling us about some of the tools that he's developed in the past, and one in particular is a tool that Akshay will show you how to use later that's called enrichR. So without further ado, we're a little bit late on our time. We'll go ahead and welcome Avi.
Yeah, thank you, Matt. I appreciate the invitation, and hi, everyone. Sorry for the delay.
All right, I hope that our mics are now working still. So let's go ahead and we can ask for a couple of comments, just a thumbs up to make sure that the audio is coming through all right. But let's go ahead and just dive right in. So first, why don't you tell us a little bit about what you do, what your lab does, and the sort of problems that you're interested in solving?
Yeah, so the lab is a computational systems biology lab. It's a dry lab, and it's really busy with doing a lot of data processing for the different data sets from the public domain, integrating them, abstracting them in a way that they can be used for data analysis and data integration. We also work with experimentalists directly analyzing the data that they collect, mostly from RNAseq and proteomics, single-cell RNAseq. And we also develop algorithms and tools and web-based tools that enable mostly experimental biologists to do analysis as well as computational biologists. So we really like to do this software development approach to research.
Okay. And one of the things that we were talking about before the audio started giving us a couple of problems was this idea about data science. So you were saying that you like to use data science and some of the tools of data science in your lab. But of course, data science is a new term, and so trying to capture a lot of different ideas within the computational biology and bioinformatics space. And so what does data science mean to you? How would you define it?
So my background before I came into biology was in computer science. So the idea is that you infuse biology with computer science concepts. And the field sort of started to move in that direction of data science. The field of molecular biology and cell biology started moving towards the idea of data science with a term called systems biology, which is now maybe less popular, but it's the same idea. Is the idea that you can build, complex networks, analyze large data sets, genome-wide data sets, to gain much more data-driven hypotheses or insights about what's happening in cells and what's happening in disease, human disease, and then you using those approaches, either statistical approaches or more topology, graphical approaches, or even dynamical modeling to understand how systems function. So this is sort of a data-driven approach to biology instead of looking at individual proteins and reading the literature and trying to figure out things one part at a time.
So one of the things that that has been said about data science is that the tools have now become more accessible than ever before. But that's a double-edged sword, so to speak, because it's giving some very powerful tools to some people who aren't trained. And so on one hand, the proponents are saying this is great because more and more people are able to use some of these data science tools to really get into their data set. But some of the voices of caution are saying, well, we need to be careful because now what we're doing is we're facilitating some analyses that might not be robust. Where do you stand on that?
Yeah, I think there is danger in giving, I guess, even the public the ability to, for example, predict drugs for diseases. So if you are, let's say, diagnosed with a disease and you start doing research and you have those tools that tell you, okay, we can prioritize drugs for your disease, and then you start taking those drugs, that's very dangerous. So I think on one hand, yes, there is a risk, but on the other hand, there is also an opportunity to enable a lot of people that it could be properly using those tools to gain much rapid discovery and understanding of the system that we're trying to figure out. So I'm like standing on both sides. I'm understanding the issues, but also I think it's important to continue like to do those type of important work, because those approaches are very powerful. I also want to add like machine learning and AI, which is also like becoming more and more commonplace. And we've been advocating for those approaches for many years now, and finally we see that people realize their power. Also, data visualization is really important, and people are becoming more and more aware of how much data visualization is critical.
Absolutely. I suspect it also makes your job slightly more difficult, because now not only do people come to you asking for help with analyses, they come to you to ask, can you look at my code to see if I did this properly?
So, yeah, I don't know, like I haven't looked at code for many years now, and the coding, doing it right, like coding wise, I think it's separate from actually understanding the problem very deeply. And we do get a lot of users that ask for help, interpreting the results. And in that particular case, it's difficult because you have to really know the biology, and you have to know the problem that you're studying very deeply in order to really gain the best insights from those results. So there is a gap, like you suggest.
Yeah, absolutely. So let's talk about one of these tools in particular, this enrichR. So why don't you tell us in your words, what exactly is enrichR?
So enrichR has first a long history, like how it became about. But in general, before I go to tell that story, enrichR is a Geneset enrichment analysis tool that basically you come in to the tool with a gene set that you identified experimentally or found somehow, and then you query enrichR with that list. And the advantage of enrichR, or the real power of enrichR, is the breadth of gene set libraries that it has. So enrichR has right now, I think 160 something gene set libraries and over 300,000 annotated gene sets. So it's like a search engine that gives you some idea about what your list contains by comparing it to those hundreds of thousands of annotated gene sets. And then you can really understand or get some clues about what is common about the genes that you came across. Yeah.
So why don't you take us back now? So what were some of the tools before enrichR, and what were the early days like?
Yes, so for my PhD, I did this project where I read biochemistry papers and extracted cell signaling direct physical interactions from those papers. So I read over 1,000 papers, and my idea was that, okay, if I read all those papers, I may understand how the cell signaling network works. And I really didn't get there, and it was also very daunting to actually read papers and extract those interactions. I didn't want to continue that. And when we started the lab, our first project was a tool called KEGG, which is kinase enrichment analysis. So at the time, there was no way to do kinase enrichment analysis. So we had a lot of data from databases that listed kinase substrate interactions. And we came up with this idea, okay, you put in your list of proteins, and then you can get the ranked list of kinases. And then that was sort of like a new way to apply enrichment analysis to data sets that no one thought of. And then we started realizing, okay, there is a lot of cheap-seq data for transcription factors. So we manually went to publications that list those targets for those transcription factors, and then created a tool called CHIA, which is like cheap-seq or cheap-X enrichment analysis. And we had those two tools, KEGG and CHIA. And then from there, it became pretty rapid that we were able to realize that there are so many other data sets that are out there in the public domain that can be converted to gene sets. And then that ended up being a tool called Lists to Networks that is no longer online. It was actually a really nice tool, but the person that developed it, he left the lab, so we could not really maintain it. And then I assigned the project to Edward Chen to maintain Lists to Networks, because we started to get a lot of users for Lists to Networks. But Ed basically said, oh, I want to rewrite it and I want to do it my way. So I let him just go ahead, and he really did a great job with the user interface and the API. In general, enrichR is a relatively simple app. It's just a database of a bunch of gene sets and some very simple tests to compare the gene sets and then to visualize the results. But there is some really advanced features there, like the fact that it's very fast is not a trivial thing. The database, the way we store the lists, it's done in a very compact way. So we have a way that the database doesn't really become too large. So, but overall, Ed did a great job, and also he did a great job in a way that he made it that other people can maintain it. So one of the key aspects of having a bioinformatics tools in a bioinformatics lab is to be able to maintain it over many years across different people that joined the lab. So there are several other people that maintained it since then and expanded it and enhanced the features of it. So it did not become that popular right away. So it was published in 2013, and the traffic grew gradually every year. And it was mostly by word of mouth that it grew. And right now we have about 1,000 users every day for enrichR. And overall, I think if we count all the users since 2013, it's already over 250,000 unique users.
Yeah, that's impressive. One of the questions that audience member wanted me to ask was, in the original enrichR paper, you described crowdsourcing some of these gene sets as was it part of a MOOC that you were teaching or running?
Yes, yeah, I can tell that in that story. So that was an exciting time of the first when Coursera was relatively in early stages. We were one of the schools that contributed MOOCs on that platform. And before they changed the way it's run, which is in an asynchronous way, the courses were staged to run at a specific time point, and they were completely free. So there was sort of a build-up of a whole lot of users, and during the course we actually communicated with the students. And at the time we asked the students, and we had I think 10,000 students that took that course, and we asked people to help us with this extraction of gene sets from Geo. And about 70 students from all around the world contributed to this crowdsourcing micro task of extracting up and down genes for signatures for drugs, gene perturbations, and disease perturbations. So we had a collection of about 5,000 signatures that we were able to add to enrichR as a source where people can query now those gene sets and find connections.
That's a really clever approach. Just like any sort of open source effort in order to gather data, one of the challenges is always quality control. And so how do you think about quality control going into enrichR for the data sets that or gene lists that could be queried?
Yes, I think it's important to not completely have no way to quality control what's coming in general. So everything I manually review it myself before we actually add something to enrichR. And the good news, or what you have to realize, is that because it's a search engine, I guess the low quality lists are never going to become enriched. So if you have something very random, or if you have a lot of like the wrong IDs, those lists will get pushed at the bottom. So it's almost like a Google search that some, I guess, not useful websites will never show up at the top. So I think in a way it sort of like takes care of itself, but also you don't want to have just a whole lot of junk there either. So we are manually reviewing every entries, and then we are careful of adding stuff that we think it's not going to be useful.
Now I'm just thinking about the future of enrichR, or the future of gene set analyses. Where do you see either enrichR going or the field going in order to try to assign functions to genes, or to find associations between genes based on gene sets or gene expression patterns? Where do you see it going?
Yes, I think still like we haven't really exploited the opportunity of the accumulated data in enrichR for doing machine learning and predicting gene function for those genes that, let's say, are understudied. So there's a lot of genes that no one knows what they do. And you could take all of the data sets in enrichR and you can basically build a machine learning platform or classifier that predicts the function of those genes that are not well annotated. You can also do this for drugs. So one of the projects that we have is called Drug enrichR or Drug Monisome, which is doing the same thing, collecting attributes about drugs, and then you can do those machine learning approaches and apply them to predict the functions of drugs for diseases, side effects, and stuff like that.
That's great. One of the things that we're trying to do with this small effort that we call Tidy Biology is to provide hands-on learning tools for people who want to learn them. Some of these computational skills and data science skills. So one of the final questions that I then would just like to ask you is, how would you then recommend some young trainees, either in graduate school or maybe a postdoc looking to retool, who wants to get more into data science and more into computational biology? What advice would you give him or her?
So I think a lot of people are shying away from programming, but I think programming is the really important skill. So, and I think in general, biologists should spend like serious effort in learning how to program, but also learning other aspects of math, statistics, computer science. And then the computer scientists and the informaticians, sometimes they feel comfortable in that space where they can do some stuff, but they don't really understand the biology. Like we had a person in the lab that was bragging about the fact that he knew only the name of one gene. So if you ask him anything a little bit in depth, and he published a bunch of papers, he did very well, but I think computer scientists and bioinformaticians should try to study as much as biology as they can, so you can be versed in both fields. So it takes a long time, you have to be patient. A lot of people want sort of like the results right away, but it takes many years to master either fields. So just realize that there is a lot to learn, and the more you know, the more powerful and more successful you're going to be.
Okay. So then we have just a couple of minutes now right at the end here for some questions coming through in the comments. So Ashley Chi asks, how do you compare and benchmark different ways of pathway enrichment?
Yeah, so we have published some ideas about how to do that. So one of the ways to do that, for example, we have data for transcription factors, and we have, let's say, ChIP-seq data, and loss of function, again, a function of a transcription factor using RNAseq or microarrays. So now you have like two independent sources of evidence about the function of a transcription factor. And even if it's in different cell type, you get some overlap. So you can take, like, let's say, one type of data, let's say the CheapSeq data, and you can use that as your training data, or you can apply your enrichment method on the CheapSeq data and then see how it ranks a transcription factor if you give it a gene set from this other data. And then you can do that repeatedly with different transcription factors, and you can flip your benchmark datasets versus your input datasets versus the gene set library, and then you can start seeing a pattern of which enrichment analysis algorithm or method beats the other one. So you don't need, it's sort of like a silver standard approach, but it's also not using all data. So it's using, using real data. So it's using the experimental data. It's not like a synthetic type of a benchmark that most people are using. So, and you can do it with other data sets like that are not necessarily transcription factor. You can do it with like drug perturbation data. You can do it with some disease GWAS type of data. So, and then you can benchmark different algorithm for enrichment analysis like that way.
Okay. We have one more question from a young graduate student, Wenchin Tong. This is a bit of a technical question. She's asking that because enrichR can do pathway analyses with or without weighting the gene, she's wondering what might be a good way to calculate the weight of gene. You know, thinking about p-values or rank. What would you say to her?
So it depends on the context, but in general, like the way it works right now in enrichR is that you want to normalize the data to have values between 0 and 1 or -1 and 1, and then you can this way it works better because this is how it computes this enrichment, this fuzzy enrichment. But there are other methods that are used when you have a ranked data set. So if you have both the gene set ranked and the input ranked, then you may use methods like cosine distance. If you have one side that is ranked, like let's say the input is ranked but the gene set is not ranked, then you can use something like GSCA, which doesn't perform that well, but so it's still like a little bit of an open space for improvement.
Okay. So then one last question from a faculty member here, Mark Herman. He asked, have you considered integrating GWAS results into enrichR? Some SNP set enrichment analyses, perhaps?
Yes, so there are already like a bunch of GWAS libraries there. We have GWAS of the UK Biobank, GWAS Catalog, dbGaP. Those are all GWAS from, they have gene sets from those GWAS, but doing Snips enrichment analysis is a good idea in general. But one of the issues with Snips, there are a lot more Snips, and it's a little bit going to be challenging because it's so sparse, to actually perform enrichment analysis at the Snips level. But we thought of it, we never got to do it, but it is it's a little bit more challenging, but it's definitely can be some useful tool. Yeah.
Okay. All right. Well, Avi, I really appreciate your taking the time to tell us about the enrichR. We're actually going to switch over now and have Akshay Bareja give us a live code along using the enrichR R package, that calls the API for your website. So thank you again. And
I really appreciate the opportunity to talk, and thank you for doing this. And I'm excited to see that you guys are using it in the course.
All right.
All right. Bye-bye.
Okay, so give me just a moment now to try to switch over and get the Akshay on the line to see if we can go ahead and start the live. All right, let's see. I think Akshay is about to sign on, so let me switch over and let's go to here. And Akshay, you are on. How are you, Akshay?
Good. Hey, Matt. How's it going?
Great. Tell us what you're going to be showing us today.
So we are going to go over how to apply enrichR to a gene expression data set, so an RNA-seq set. And we'll pick a pathway of interest and then visualize that pathway using the PathView package.
Okay. Tell us where your data set comes from today.
So this data set is based on a real, actual RNA-seq data set out of Jim White's lab. Shouldn't have said that, but it's been anonymized, and the numbers have been changed a little bit. There's not really a huge amount to say that's biologically interesting, other than I made two comparisons: control and treatment.
Okay.
But the rich data set, there are many genes that I've compared, so it should hopefully yield something interesting.
Okay, so you'll show us the enrichR R package today, and what else will you show us?
This package called PathView, and what that allows you to do is it allows you to annotate KEGG pathways, so that you can get, for example, so we'll be looking at the insulin signaling pathway, so we can get that pathway sketched out with genes that are either up or down regulated, colored in.
Okay, let's dive right in. Go ahead.
Sorry, I just need to be reminded of how to share my screen.
Look for the button that says share screen. So in your lower right-hand side, there should be two windows sort of stacked on top of each other. That's the share screen icon.
Can you see my screen now?
Yes, we can.
Excellent. All right. So before we begin, make sure that so there are a number of packages we'll need to use. Matt, apparently people can't hear me.
Mark just said that too. So let me try a couple of things here. No, that's now we see your YouTube window.
Oops, sorry.
Okay. Yeah, this is strange. This is what happened with Avi in the first stream. Okay, yes, sound is back on, Mark says. Okay, so go ahead and try it again.
Okay, so can people hear me now?
Yes, I believe they can.
Okay, excellent. So I guess I'll start from the beginning. So for people who've just joined, make sure that you have the Tidy Biology Meetup project open. And to make sure that it's up to date, go to your Git pane and click this blue arrow. Mine is already up to date. Once all of that is done, go to the folder marked 2020_05. And in it, click on the code folder. And in that code folder, you'll see an RMD document called enrichR.rmd. So go ahead and open that. Now you'll see in the first chunk, there are a number of packages that I'm going to assume you don't have installed. So the code to install it has been commented out. So all you need to do is uncomment it and then run the whole chunk. Now, all right, so I'm going to go ahead and, the most important lines 13 and 14, because some of these packages will be downloaded from Bioconductor. And to install packages from Bioconductor, you need to have this BioC manager package. Now, I'm just going to go ahead and reinstall one of the packages just because it takes a little while. And there's this slightly interesting thing with Bioconductor packages that could be problematic. So if you don't have your console open, like I do here, what you'll see is it'll look like something's happening, and something is happening, but it will look like something is stalled. So you have to have your console open so that you can see how things are progressing. This is important because if your console is closed, eventually it will ask you to choose an option. And that might not be clear if your console is not open. So this is almost done. So it'll ask you to update all, some, or none of these packages. So I just go ahead and update none of them, and that should be fine. And do this for all the other packages. And then go ahead and execute the library command for all of them. This will take a few seconds. So while this is going on, I'll talk a little bit more about the dataset we'll be using. As I mentioned, it's an RNAseq dataset, and it's just comparing a control with a treatment. It has a log fold change, columns for log fold change, and the adjusted p-values. And we'll be filtering based on filtering so that we get a set of upregulated genes. And then we'll do pathway analysis on those genes. So this is nearly done. Great, done. So the first thing we'll do next is import this data set. So it's on line 26. So we run this, and let's take a look at this data set. So if you go to environment, as I just clicked on that, you'll see data up there along with a couple of other data sets that came with one of the packages we just installed. So we'll run the dim function, which, as you remember, is a very simple function that gives you a very simple output. So it gives you the number of rows. So we have 4,287 genes and number of columns. We have 5 columns. So we can take a quick look at what those columns are by running the glimpse function. And we see we have one character variable, that's the gene name, and we have 4 doubles. So those contain numeric values. And we can view our data set by running the view command so that we can see the data set in a different, open up in a different pane here. And again, you see this is a pretty familiar looking data set. All right. So now let's take a look at the enrichR package itself. So we can, if you go to line 38 and run the lsf.str function, you can see down here that the enrichR package is a really lean one, and it only has 3 functions. enrichR, which is the actual function we'll be using to do the pathway analysis. The list enrichR DBS, which gives you a list of all the databases that are included in this package, and the print enrich function, which will print the output to a tab file, which we won't do that. So let's go ahead and run the list enrich DBS function on line 39. And I like that Avi compared this to a search engine. So what we can do is, so if you click on DBS, or actually if you view it, you can see all the libraries that we could potentially query. And if you click on num terms, you can see some of them are as small as contain only 12 terms, and some of them contain 33,000 terms. So you can go through that the DBS data frame to pick your data frame of interest. So for today's walkthrough meetup, we'll just select these 3 databases: the KEGG 2019 mouse, Go molecular function, and Go biological process. And so what we do is we make a character vector called databases. So I'll just go ahead and run that. and it just contains the names of these 3 databases. What we'll then do is, if we go to line 44, we can filter our data set called data so that we only have terms that have a log fold change greater than 0. So we're only interested in genes whose expression has been upregulated. And then we use this pull function to only obtain the names of these genes. And we can save that as upregulated. Now, upregulated is, like databases, is also a character vector. So you can go ahead and run those 3 lines. And in fact, if I just run upregulated in the console, you'll see it's a big list of gene names. So to run the enrichR function is, so we'll be doing it in without any waiting. It's really simple. The enrichR function just takes 2 arguments. One is the list of your genes of interest and a list of the databases. So we'll go ahead and run line 48. And it'll take a couple of seconds to run. Done. And if you click on enriched up, which is how we've saved this, you can see that there are slots for each database queried. And since we queried 3 databases, we have 3 slots. So we'll go ahead and just look at the KEGG 2019 mouse database. So if you run 51, line 51, you'll just see the top 10 terms. And yeah, so these are ordered in descending order of significance. And you can see the term itself, the overlap, so that's the number of genes in your data set compared to the number of genes in the specific term. You have your p-value, your adjusted p-value, and you can see these are all really low adjusted p-values, which is great. And if we scroll to the right, eventually we'll reach, in the genes column, you'll see a list of genes that are within that specific term. And this is great for, if we were to stop now, we'd already find out a lot of information about pathways that are overrepresented in our data set. Now, if we want to delve into one of these pathways, one way is to visualize this pathway. So let's take the, I'm arbitrarily choosing the insulin signaling pathway. Let's say that's the pathway we're interested in. We can go ahead and visualize that pathway using the path view package. So if you go to line 56, we'll do the same thing. We'll take our data frame. We'll filter it so that the terms are only upregulated, so log 2 fold change over 0. And then what we need to do for path view to work, so actually if we go back to data, you'll see we have a gene name column. Now the path view package will not accept gene names. It needs to have entree IDs. So to do that, we can convert the gene names to the entree IDs. And the way we do that is to use this package that we downloaded from Bioconductor called org.mm.eg.db. And we can use the mutate function on line 58 to create a new variable called gene ID that contains the corresponding entree IDs for the gene names. And then what we do is, in case there are any missing matches, we'll use this drop NA function to just drop those genes. So let's go ahead and run this. And we save the output of this in a data frame called upregulated with ID. Now, there are a couple of uncommented lines, commented lines here on 61 and 62. So go ahead and uncomment them. I'm just going to do this because doing it this way results in an error. And I thought it would be interesting to show what that error is and how to troubleshoot. So what we want to do is again, to prepare the data set that is needed, that is necessary to do path view analysis, the first thing we do is make a data frame with just one column, and that just contains the fold changes for the genes of interest. So again, we'll just do upregulated with ID, pipe it into select, and select the log 2 fold changes, and save it as a data frame called FC. You will notice that on line 62, instead of just running the select function, I did dplyr : select. And the reason I did that is if there's a conflict between select within the dplyr package and a select that's used by one of these bioconductor packages. So you have to explicitly state that you're using select from dplyr. So let's take a look at fc. And it's simple, just one variable with all these log2 fold changes. To this, we need to now give each row a row name, and that row name is the entree ID for the corresponding gene. So to do that, we can just use this function called row names for the data frame we just made, fc, and then extract the gene ID column from the upregulated with ID data frame. So we can go ahead and run that. Now, if we run that, we come across a warning and an error. So the warning is that the tidyverse doesn't like giving rows names. Columns can have names, columns must have names, but rows, they frown on that, but we can ignore that for now. What we can't ignore is the error, and the error is here: duplicate row names are not allowed. So this means that there's at least one gene that appears twice in our data set, and we need to find out what that gene or genes are. So one way to do it is to see the number of genes you have. So if you look at line 65, we can use the length function to see the number of genes we have. So it's 2,146. Now we can compare that to the number of unique gene names we have. So to do that, it's on line 66, and it's identical to 65, except we also use the unique function. And you'll see we have 2,145, which means there's one term that's been duplicated that appears twice. So let's find out what that term is. So if you go to line 68, we can use this duplicated function to identify any duplicates in our data set. So the way the duplicated function works is it takes a list of entries and returns a false if there are no duplicates and a true if there are. So if we were to just run this, so I'm just going to copy this duplicated function. If we were to just run this, we'll get a bunch of falses. That's because most of the genes, nearly all the genes here are not duplicated except for one. So what we can do is we can use all these falses and one true to subset our upregulated with ID data frame. So we can use this duplicated function output in the row portion, and then we can select all the columns. So we can go ahead and run this. If we scroll down, we'll see that it's the gene that appears twice is MICAL3. And to confirm that this is in fact a duplicate, we can go back to our data frame and then just do a search for MICAL3, and you see that it does appear twice. So one solution to this is to just drop the second entry, which I feel comfortable doing just because the log2 fold changes are very similar between these two terms. All right, so that was a bit of a detour just to do some troubleshooting. And if you go back to line 72, it's very similar to the code we ran previously, except what we do is we use this function on line 76 called distinct, and the term we're applying this distinct function to is gene ID. So as the name suggests, we only want a distinct output. So we don't want any duplicate output. So we know that this will result in a data frame that drops the second entry of the MICAL3 gene. And then it's essential that we set this .keep_all argument to true, because if we don't do that, we'll just end up with a data frame with one column. So we go ahead and run that, and we get a new upregulated with ID data frame. And actually, we can just take a look at that. So that's very similar to a data frame, except we now have this new column called gene_ID. So we go ahead and then rerun the code here, so 77 and 78, so that again we get a data frame called FC, which is one column that has the log2 fold change values. And if we go to line 80, we can give each row a name, and that name is the relevant gene ID. So run line 80. If we click on FC, you'll see now, in addition to a column of log2 fold changes, you have each row that has its own name, and that name is the entree ID. Great. So all of that has been done to prepare a data frame that is suitable for path view analysis. So line 83 is the line you need to run to do this analysis, and the function is called path view. And the arguments are, you need to give it a data set in the right format, so that's our FC. You need to give it a pathway ID. So in this case, as I mentioned, we will be looking at the insulin signaling pathway. Now, to find out the relevant ID, just go to the Kyoto Encyclopedia of Genes and Genomes website and search for insulin signaling. And it'll give you a list of Go terms with the corresponding pathway IDs. And we want to set the species to mouse because this is mouse. And this out.suffix upregulated just adds a name to the PNG file that will be generated and saved in your working directory. So I'm actually not going to run this line. You can run this line. I'm not going to do this because I've already saved the output in a different folder called results. So I'll just show you what that output looks like. So if you run line 86, you can use the read image function from the EBA image package to load that, and then just run the display function on line 87 to see what the pathway looks like. So we can pop this out to get a higher resolution image. And you see the insulin signaling pathway, and you see a number of genes, all of whose expression is increased. And so this corresponds nicely with the enrichR output, and it allows you to gain a better sense of where those genes whose expression has been upregulated, where they lie in this insulin signaling pathway. Yeah, that's it. So that's how to use the enrichR package to identify pathways of interest, and how to use the path view package to visualize those pathways. All right, back to you, Matt.
All right, thanks, Akshay. Okay, and thank you all for another Tidy Biology meetup. We had some technical difficulties, but we made our way through it. So join us next month, last Monday of June. We'll be back here. So I hope to see you then. Bye-bye.